Hello everyone, hope you are doing well. I am Rafiah Kanwal and I will be presenting metastasis in renal cell carcinoma, biology and treatment. I will be discussing at first the abstract, then introduction, and then aims and objectives, renal cell carcinoma, biology, treatment, and at the end conclusion. So let's begin with the presentation. Renal cell carcinoma is a heterogeneous group of cancer that reacts inappropriately to recent therapeutic methods and is frequently linked with uncertain clinical channels. Heterogeneity is due to distinct cellular morphology and phenotypic profiles. Biology of renal cell carcinoma depends on histology, tumor diversity, and biological mechanism of metastasis. Histology is a branch of biology that is concerned with the composition and structure of tissues in relation to their specialized functions. Tumor shows diversity in gene expression and drug sensitivity. Treatment has been transformed from cytokine-based methods to targeted agent therapy against vascular endothelial growth factor and most recently to immunotherapy drugs. Before discussing the biology, mechanism, and treatment, I want to introduce the renal cell carcinoma. It encloses a diversified class of cancer attained from renal tubules epithelial cells. Renal tubular epithelial cells lines the renal tubules and is one of the 10 prevalent cancers worldwide. Radical nephrectomy vision was removing the entire kidney along with a section of the tube leading to the bladder, the gland that sits atop of the kidney, and the fatty tissues surrounding the kidney. It can treat localized renal cell carcinoma. However, almost 30% of localized RCC patients ultimately develop metastasis that cannot be treated with radical nephrectomy. And it requires systemic therapies which focuses on the interactions and relationships between groups and is linked with high death rate. When metastasis arises in RCC, it usually is suppressed to bone, brain, pancreas, and renal glands, gallbladder, liver, and lymph nodes. Patients with metastasis RCC survive up to 8 months, with 50% dying within 1 year. Immunotherapy with cytokine was previously used as a quality treatment. Recent therapies used for metastatic RCC involve targeted therapies, tyrosine kinase inhibitor. Some current approaches are anti-MET and anti-RET. Finally, chemotherapies including PD-1, anti-PD-L1, and anti-CTLA-4. Targeted drugs against vascular endothelial growth factor and mammalian target of rapamycin have shown effective results and progression-free survival. Vascular endothelial growth factors promote angiogenesis. While mTOR, which is mammalian target of rapamycin signaling, regulates cell proliferation and metabolism, involved in tumor initiation and progression. Moving further to the aims and objectives of this topic is to summarize the biology and molecular mechanism of renal cell carcinoma and to understand the present and the future directions in the treatment of RCC metastasis. Now let's discuss the biology of RCC metastasis in order to understand different treatment options available. Almost 70% of patients with clear cell renal cell carcinoma endure mutation in the von Hippel-Lindau gene. Wild-type von Hippel-Lindau gene acts as a ubiquitin ligase and negatively regulates the expression of a large collection of hypoxia-inducible genes. That is controlled by hypoxia-inducible factors. Wild-type VHL causes the disintegration of hypoxia-inducible factors. And in patients enduring the von Hippel-Lindau gene mutation, the subsequent rise of HIF level increases the level of vascular endothelial growth factor. And HIF regulates the several genes including vascular endothelial growth factor. High levels of HIF leads to high level of vascular endothelial growth factor, and this ultimately activates the VEGF receptor, thus activating the phosphoinositol 3 kinase AKT signaling cascade. The mammalian target of rapamycin is triggered downstream, causing the transcription of numerous tumor-promoting factors. Consequently, increases the cellular migration. Several tumor-promoting factors are transcribed. These include These include the fibroblast growth factor, platelet-derived growth factor, vascular endothelial growth factor. Other transmembrane receptors also play a vital role in driving the metastasis, such as MET and AXL. MET is a mesenchymal epithelial transition, is a reversible biological process that involves the transition from motile multipolar mesenchymal cells to planar arrays of polarized cells, called epithelia, and shown to occur in cancer metastasis. MET encode unconventional receptor tyrosine kinases with pleiotropic functions. While AXL is a member of TAM family with the high affinity ligand growth arrest specific protein that is GAS6, the GAS6 AXL signaling pathway is associated with tumor cell growth and metastasis, angiogenesis. Papillary RCC is a cancer of the tubes that filter waste products that are removed by kidney from the blood. There are two types of papillary renal cell carcinoma. Type 1 is most common and grows slowly, while the type 2 is more aggressive and grows more quickly. Papillary RCC has two subtypes. While SETD2 is a protein that codes gene coding, it's a protein coding gene containing histone, lysine, methyltransferases. SETD2 plays an important role in the initiation, progression, prognosis, and treatment of solid tumors. SETD2 participates in transcriptional regulation, DNA mismatch repair, and some other processes that ultimately leads to the progression of malignant tumors. While CKDN2 is a cyclin-dependent kinase inhibitor 2A, is a protein coding gene. The gene codes for two proteins, including the INK4 family member P16 and P14. While TFE3 is a transcription factor binding to IGHM enhances 3 is a protein coding gene. It encodes the transcription factor involved in the regulation of many proteins implicated in carcinogenesis, including TGF, MET, and E-cadherin. While chromophobe RCC is a rare type of kidney cancer that forms in the cells lining the small tubules in the kidney, chromophobe RCC suggests recurrent alteration in the third promoter area. Picture here shows the mechanism of RCC metastasis. There are certain changes appear in renal cancer cell. This is the renal cancer cell, and it is displayed with CMET and AXL. These are CMET and AXL, and that are both involved in tumorigenesis. Due to mutation in the von Hippel-Lindau gene, subsequent rise in HIF level increases the level of vascular endothelial growth factor that binds to VEGF receptor on the renal cancer cells. MET binds to the AGF and promotes RAS-JAF pathway and RAS phosphoinositol 3 kinase pathway. A signaling cascade starts when RAS converted to RAS, and then to MEC, and then to ERK, and this ultimately goes inside the cell nucleus and binds to certain transcription factors that results in the cell growth and survival. And the second pathway that is activated from RAS is the formation of phosphoinositol 3 kinase that converts to the AKT and then to the mTOR. These result in the transcription of certain genes resulting in FGF, PDGF, and VEGF. These bind to certain receptors on the blood vessels, and these receptors are fibroblast growth factor receptor, platelet-derived growth factor receptor, and vascular endothelial growth factor receptors. After binding of the transcription factor to the respective receptors on the blood vessel, it promotes angiogenesis. That leads to the spread of cancer to other organs and promotes metastasis. PD-L1 expressed on the renal cancer cell binds to the PD-1 on the activated T cell, resulting T cell receptor binds to major histocompatibility complex present on antigen presenting cell. While CTLA-4 binds to major histocompatibility complex present on antigen presenting cell. CTLA-4 binds to the CD80 and CD86 present on antigen presenting cell, and CTLA-4 binds to CD80 and 86 with high affinity and avidity than does with the CD80, preventing conventional T cell stimulation through CD80-CD86 interactions with CD28. Removal of CD80 and CD86 ligands by trans endocytosis result in impaired co-stimulation of T cells via CD28, resulting in immune regulation. But in this case, there will be no immune regulation as tumor development occurs, leading to the renal cell carcinoma metastasis. Actually, PD1 binds to PD-L1 and receives positive signal to T cell receptor and CD28. These immunosuppressive molecules function as brakes to regulate the adaptive immune response. This suppression of immune signal actually causes metastasis. While T cell uses their surface receptors to recognize peptides presented by major histocompatibility complex, and the peptides presented by MHC is derived from the host system as a new protein is formed due to mutations in the gene, thus termed as neoantigens. Neoantigens are basically the new proteins that form on the cancer cell when certain mutations occur in the tumor DNA. Here's the molecular mechanism of renal cell carcinoma. Microvesicles bearing CD105 positive cells carry the phenotype of cancer stem cells. Tumor-derived microvesicles released from CD105 positive human renal cancer stem cells were shown to stimulate angiogenesis. Also, the microRNA released from such cancer cells, the angiogenesis leads to spread of tumor and cancer cells to other sites. Some other factors also promote angiogenesis. One of them is the suppression of pulmonary metastasis of renal cell carcinoma. That is due to the loss of neutrophil chemokines in lungs. Chemokines are a large family of small secreted proteins. They signal through cell surface G protein. Cell surface G protein coupled receptors. They are the best known for the ability to stimulate the migration of cells, most notably of white blood cells. Chemokines produced by neutrophil in inflamed tissues amplify and polarize the immune response, and the expression of CCR7 by activated neutrophil promotes the migration to lymph nodes, where they can directly act as antigen-presenting cells. The suppression will not activate immune system and do not suppress tumor, leading to metastasis. Immune cell suppression with anti-tumor immunity causes the development of RCC due to the angiogenic effects. Mostly clear cell renal cell carcinoma cases have chemotrypsis of chromosome 3p. Chromotrypsis is a complex chromosomal rearrangement and is characterized by up to thousands of cluster chromosomal rearrangements that occur simultaneously and are localized in limited regions of the genome in one or several chromosomes. Chromotrypsis of chromosome 3p leads to the loss of 3p and ultimately inactivation of von Hippel-Lindau disease tumor stress protein. It will also initiate the angiogenesis process and invasion as it increases the level of VEGF by increasing HIF. In RCC, VHL protein complex becomes defective and do not play its role. Consequently, HIF alpha accumulates in cells. It stimulates the gene responsible for hypoxia, such as VEGF, and some others, such as fibroblast growth factor, platelet-derived growth factors, receptors, and AXL and CMET involved in angiogenesis. These factors and receptors are utilized by different factors that bind to them at the blood vessel and promote angiogenesis. Apart from its mTOR pathway started from RAS have an equally significant role in tumorigenesis by the loss of p53 expression or mutated phosphoinositol 3 kinase and PTEN genes. p53 is a tumor suppressor gene that stops the formation of tumor, but loss of p53 promotes tumor formation. PI3 kinase involved in cellular functions such as cell growth, proliferation, and differentiation that is involved in cancer. PTEN genes provides the instructions for making an enzyme that is found in almost all tissues in the body. The enzymes act as a tumor suppressor, which means that it helps regulate cell division by keeping cells from growing and dividing too rapidly or in uncontrolled way. Now coming to the RCC treatment, FDA approved many anti-angiogenic drugs, including sorafenib, sunitinib, pazopanib, exitinib, bevacizumab, and cabozantinib. Bevacizumab is a targeted cancer drug treatment, also known as Avastin. It is given by slow injection into a vein. It is directed against VEGF. Cabozantinib is a class of medication called kinase inhibitor, and it is sold under brand name Comtrix. It is inhibitor of MET, VEGFR, and also inhibitor of AXL and RET. Sorafenib, sunitinib, pazopanib, and exitinib is an inhibitor of VEGFR and PDGFR. Treatments to different drugs are selected on the criteria of individual patient factor. For example, patients with active immune disorders are not given the checkpoint inhibitor nivolumab. Nivolumab can worsen pre-existing autoimmune diseases. Sunitinib and pazopanib widely used as a first-line agent due to their association with the high response rates and development-free survival. In a study of pazopanib versus sunitinib, pazopanib was superior to sunitinib in terms of progression-free survival. Surgery plays a significant part in the treatment of metastatic RCC. In solitary metastasis, the metastatic foci are surgically resected as a treatment option. Atezolizumab and nivolumab target PD-L1 and PD-1 respectively, and ipilimumab targets CD80 and CTLA4. And hence causes the immune activation. Similarly, bevacizumab was used against VEGF, nivolumab against FGFR, and everolimus along with the temsirolimus against mTOR. Moreover, cabozantinib targets multiple proteins including c-MET, AXL, and VEGFR. Furthermore, multiple drugs can target PDGFR and VEGFR, including sunitinib, pazopanib, exitinib, lavatinib, and sorafenib. Nivolumab combined with ipilimumab was found to be more effective than sunitinib and showed better health and quality of life. Different drugs show different extents of better health quality despite the undoubtable advantages of immune checkpoint inhibitors in treating metastatic RCC patients. Combination therapy is found to be more effective. Combining anti-VEGF agents and immune checkpoints is an effective substitute that can overcome the resistance of immunotherapy and can direct both against angiogenesis and tumor microenvironment. Immune checkpoint inhibitors include atezolizumab used against PD-L1, anti-VEGFR, sunitinib, pazopanib, sorafenib, and patients showed better treatment survival with results with the combined therapies anti-VEGF and anti-PD-L1 that is more effective. The picture here shows the targeted therapies that are used as inhibitor. Atezolizumab is inhibitor of PD-L1, and nivolumab is inhibitor of PD-1. This will release the suppression of immune system. Similarly, immune activation results by delivering ipilimumab against CTLA4. These are immune checkpoint inhibitors. Other targeted drugs include bevacizumab that is delivered against VEGF that stops binding to VEGFR and prevents angiogenesis. mTOR formation is inhibited by everolimus and temsirolimus. mTOR pathway involved in angiogenesis process. By inhibiting its metastasis can be prevented. Moreover, c-MET and AXL along with VEGFR is inhibited by cabozantinib. As c-MET involves in tumor formation and AXL involved in metastasis and angiogenesis, their inhibition will suppress tumorigenesis and prevent cancer spread. Many drugs have been used against PDGFR and VEGFR, include sunitinib, pazopanib, exitinib, lavatinib, and sorafenib, and their inhibition prevent angiogenesis process. Lastly, lenvatinib is used against the fibroblast growth factor receptor and also inhibit angiogenesis. Finally, towards concluding my presentation, renal cell carcinoma is a diversified category of cancer that is prevalent worldwide. Different receptors are involved in metastatic RCC, including VEGFR and FGFR. Proteins such as FGF and VEGF expressed because of mutated VHL that leads to the metastasis. Targeted drugs involved against mTOR and VEGF showed much better outcomes. That's all from my side. Thank you. These are the references.